Good afternoon. You're welcome to my channel. I remain Dr. Uduak. Today we are going to talk about an important topic which is known as taste sensation or the physiology of taste. The physiology of taste. So get ready as we begin to talk about it. When we talk about taste, we are talking about the sense of, in fact, we call it, if we don't call it taste, we call it gustation. If you don't call it taste, it is called gustation. Gustation is also known as taste, and it is very important. Now, the sense organ for taste is known as taste bud. The sense organ for taste is known as taste bud. Very important. And we usually have taste buds in the papilla. There's what we call the papilla of the tongue. And then we have taste buds in other places like in the mucosa of the pharynx. We have taste buds in the mucosa of the epiglottis. We have taste buds in the mucosa of the hard palate. And then we also have taste buds in the upper part of the esophagus, that is the proximal part of the esophagus. So it is very important we know this. And then it is also important to know that adults usually have about 10,000 taste buds, whereas children have more taste buds than adults. Very important, very, very important to know. And then it is also important to know that the diameter of taste bud is about, let's say, 50 to 70 micron. 50 to 70 micron. And then as we progress in age, our sensitivity for taste begin to decrease or wane. And then it is also important that over the years, as we advance in age, our taste buds begin to degenerate. Very important. Very important. Now let's talk about the situation of taste buds. Now, like I told you earlier on, taste buds are actually situated in the papilla of the tongue. They are situated in the papilla of the tongue. That is, majority of the taste buds are located in the papilla of the tongue. And then some taste buds are also found in the epiglottis. Some are found in the pharynx. Some are found in the upper part of the esophagus, the proximal part of the esophagus. It is very important to know that. Then let's talk about the papilla itself, the papilla of the tongue. The papilla usually contains a good number of taste buds, and we usually have three basic types of papilla. I will mention four. The first one I will mention is known as the filiform papilla, which is conical in shape. It is located in the dorsal part, in the dorsum of the tongue. It is located in the dorsum of the tongue. Then we have the foliate papilla. The foliate papilla is located in the lateral aspect of the tongue. And then we also have what we call the fungiform papilla. The fungiform papilla is located in the anterior part of the tongue, especially around the tip of the tongue. And then we also have what we call the circumvallate papilla. The circumvallate papilla is a V-shaped structure that is located in the posterior aspects of the tongue. Very important. Let me repeat myself. Let me repeat it again so that you grab the concept. The first one is the first one I mentioned is the the filiform papilla, which is conical in shape, and then, like I said, it is located in the dorsum of the tongue. Then the next one is known as the foliate papilla, that is located in the lateral aspect of the tongue, and then we have the fungiform papilla that is located in the anterior aspect of the tongue, or anterior part of the tongue, especially around the tip of the tongue. And then we also have the circumvallate papilla. The circumvallate papilla is a V-shaped structure that is located around the posterior part of the tongue. Very important. Now let's talk about the structure of taste buds. It is very important to know that about 40 cells make up the we have about 40 cells that make up the taste bud, and then these cells are divided into 4 groups. The first one is the type 1 cell, the second one is the type 2 cells, the third one is the type 3 cells, and then we have the type 4 cell. The type 1 cell is also known as a supporting cell. The function excuse me excuse me the type 1 cells are known as the supporting cells. The functions of type 2 cells are unknown. The functions of type 2 cells are unknown, or is unknown. And then type 3 cells are known as the receptor cells. Type 3 cells are the receptor cells. And then we have the type 4 cells. The type 4 cells are known as basal cells. So let me take it again. We have more than 40 cells that make up taste bud, and then these 40 cells are divided into 4 groups. The first one is type 1 cell. We have the type 2 cells, we have the type 3 cells, and then we have the type 4 cells. The type 1 cells, like I said, are supporting cells. The type 2 cells, the function of type 2 cell is unknown. And then we have the type 3 cells, which is known as the receptor cells. And then we have the type 4 cell, which is known as the basal cells. Now, the lifespan of the cells in the taste buds is around, let's say, 4 to 10 days, right? That's the lifespan. So when the taste buds begin to die, then they are usually replaced by the type 4 cells, which are the basal cells. So it is important to know that. Let me take this again, so that let me mention the types of cells that make up the taste buds, so that you grab you fully grab the concept. The first one, the type 1 cells, which are known as the supporting cells. The second one is the type 2 cells, of which I mentioned that the function is unknown. And then we have the type 3 cells. The type 3 cells are known as the receptor cells, and then we have the type 4 cells. The type 4 cells are known as the basal cells that usually replaces dead cells. Now let us talk about the taste pathway. Let's talk about the taste pathway. It is very important to know that the taste pathway is a very important concept, and a very simple concept that I believe that you should know. Now, the nerves that actually innervate the tongue, the nerves that innervate the tongues are actually the facial nerve, the glossopharyngeal nerve, and then the vagus nerve. Now, the chorda tympani of the facial nerve usually innervates the anterior 2/3 of the tongue, right? The chorda tympani of the facial nerve usually innervates the anterior 2/3 of the tongue, whereas the glossopharyngeal nerve usually innervates the posterior 1/3 of the tongue. And then the vagus nerve innervates every other aspect of the oral cavity that is involved in taste, like the palate, the esophagus, that is the proximal part of the esophagus, and then also I think I mentioned the epiglottis, the esophagus, and then the palate and the pharynx. These areas are usually innervated by the vagus nerve. Now, let's start from the first one, which I mentioned. The chorda tympani of the facial nerve, like I said, innervates the anterior two-thirds of the tongue, and then it extends down to what is referred to as the geniculate ganglion. From the geniculate ganglion, it goes down to the medulla, the gustatory aspect of the medulla, which is known as the tractus solitarius. Now, secondly, the glossopharyngeal nerve arises from the posterior one-third of the tongue and then moves down to what is referred to as the petrosal ganglion. Right? It goes down, it passes through the petrosal ganglion and then enters the gustatory area of the brain, which is known as the medulla. And the gustatory area is the tractus solitarius. And then the vagus nerve arises from other parts of the oral cavity that are involved in taste, and then passes through what is referred to as the nodus ganglion. It passes through the nodus ganglion and then goes down to the tractus solitarius in the medulla. Now, from the tractus solitarius, we have what we call the medial lemniscus. The medial lemniscus. The medial lemniscus arises from the tractus solitarius and then goes down to the thalamus. And then from the thalamus, we have the third order neurons going down to the brain. Now, the first order neuron, it is important to know that the first order neuron arises from these three cranial nerves which I've mentioned, which are the facial nerve, the glossopharyngeal nerve, and the vagus nerve. The second order neuron arises from the tractus solitarius. The tractus solitarius is found in the medulla. And then the third order neuron actually arises from the thalamus, particularly the VPM, the ventral posteromedial nucleus of the thalamus. And then from there, it goes to the parietal lobe, and then of course from there it extends down to the taste, the areas of the brain that is involved in taste, like the operculum, right? The operculum, very important to know, the operculum. Now, let's talk about taste sensations. Usually, based on the chemicals that are present in food, taste sensation is divided into 5. We normally have 5 primary taste sensations, which are: number 1 is salty. We have sour, we have salty taste, we have sweet taste, we have bitter taste, and then we have umami. We have sour taste, we have salty taste, we have sweet taste, we have bitter taste, and then we have umami. Now, the sour taste is actually it is mediated by acidic food. Yes, it is mediated by acidic food, and then of course in acidic food we have what we call the protons. Right? Then the salty taste is mediated by ions such as sodium ion and potassium ion, and then the sweet and the bitter taste is mediated by organic substance. For instance, the sweet taste is mediated by substances like sugar, alcohol, ketones, and aldehyde. And then the bitter taste is mediated by what is referred to as alkaloid substances, like organic substances like alkaloid, and the bitter taste has a very, very high it has a very, very low threshold. The bitter taste has a very, very low threshold. Then the next one is known as the umami. Umami is a Japanese word that means pleasant tastes. Umami is a Japanese word that means pleasant taste, and it is mediated by a substance that is known as the L-glutamate. L-glutamate, very important. Now, let us talk about sensory transduction of taste. Sensory transduction. sensory transduction of taste. Now, when we talk about sensory transduction, we mention um, the mechanism that is involved in salt taste and also the mechanism that is involved in sour taste, and then we now mention the mechanism that is involved in sweet, bitter, and umami taste. Sweet, bitter, and umami taste. Now, for the for the sour taste and the salty taste, it is important to know that when the receptor cells are actually stimulated, in the receptor cells we have what we call vesicles. These vesicles have neurotransmitters. When the receptor cells are actually stimulated by let's say by, for instance, if you eat a salty food and the receptor cells are stimulated, when they are stimulated, the vesicles tend to move down towards the membrane, and then when they move down to the membrane, of course the vesicles will rupture, and then the neurotransmitters will be released, and then of course it will bind to the nerves. It will bind to the neurons or the nerves that is involved in taste. And then when that happens, of course, you know that receptor potentials will be generated. And now the more the stimuli, the more the intensity of the stimuli, the more the amplitude of the receptor potential that is generated. And the more the amplitude of the receptor potential that is generated, the more the action potential that is generated. Now, for salty food, or for let's say for salty food. Now, when you eat when you take something that is a food that is salty, you observe that actually there are some receptors that are stimulated, which are, of course, they are ionotropic receptors. Now, the salty food tends to stimulate these ionotropic receptors, and then they tend to the salts such as let's say sodium ion, right. Sodium ion tends to bind with a special channel that is known as what we call the epithelial sodium channels. When these epithelial sodium channels are stimulated, it allows for the opening of these channels. When these channels open, it allows for the influx of sodium. When sodium gets into the receptors, what happens is that it blocks potassium channels, and when potassium channels is blocked, it will now result in the depolarization. It will result in the depolarization of the receptor, and then once there is depolarization of the receptor, it will result in the generation of receptor potential. When receptor potential is generated, it will result in the generation of action potential. Now, of course, you know that the receptor potentials are graded potentials. The more the intensity of the stimuli, the more the intensity of the amplitude. The more the amplitude of the receptor potential, and then of course the more the amplitude of the receptor potential, the more the action potential. So that is very important. Now, when you take in sour food, you observe that a proton like hydrogen ion will bind to the epithelial sodium ion. The epithelial sodium channels, which will result in the influx of sodium ion, and then which will result in the influx of hydrogen ion, that will result in it will block the potassium ions. Right? It will block the potassium channels and prevent the efflux of potassium ion. And when that happens, it will result in the generation of receptor potential, which will result in depolarization of the receptor. And of course, like I mentioned earlier, that the more the intensity of the stimuli, right, the more the amplitude of the receptor potential, and the more the amplitude of the receptor potential, the more the action potential. Very important. Now let us talk about the medical implication in taste. The first one is ageusia. Now, ageusia actually means loss of sensation. Now, the second one is known as dysgeusia. Dysgeusia actually means disturbance in taste sensation, which actually occurs during what is referred to as temporal lobe syndrome. And then we also have taste blindness. Taste blindness is a rare genetic disorder in which an individual fails to recognize substances, by taste, or we can call it loss of taste as well. Then we have hypogeusia. Hypogeusia is also known as decrease in taste sensation. Thank you for listening. I hope to see you in my next class. Thank you.